and I have additional locality data, or where your find was made. You should record that data in a way that if you handed it off to someone, they could follow your directions and reach your site, if you're interested in doing it. Okay. Okay, so for the scat, you found your scat, you documented your scat. Sometimes, depending on how old the scat is, if it's more than 72 hours, you're probably not going to be able to get viable DNA from it. You can, using a spray bottle and sterile water, you can rehydrate it. Then with gloved hands, because obviously you don't want to contaminate it, so the lab says, oh, it's Homo sapien. And you know damn well no one went out there and took a crap, because it's your DNA they're reading, because you collected it wrong. You can use, I have in this box here, it's a little Plano plastic box that I've designated my DNA collection kit. And I've got labeled envelopes for hair specimens with labels with that information I've been talking about on them. Magnifying glasses, notebooks, and then I've got DNA collection for scat. And this is 100%, it's Everclear. Now you're going to want to check also with open container laws if you're riding around with this in your truck and you tell a state trooper that you're collecting Bigfoot crap. I imagine
you're going to blow.
Yeah, you're going to blow. It's going to have a laugh and you're going to have a ticket. But hey, to hell with it, because you got some Bigfoot DNA, right? Here in this little baggie, I've got everything separated so it's easy to get. Obviously, these are the gloves. Gloves you're going to want to use to prevent cross-contamination, because how many stories have you heard of someone finding something, sending it in to a lab, they perform their downstream analysis, and it comes back as, you know, it's human. Okay, so something as simple as gloves, that can help prevent that. The face mask would also come in handy to prevent you from sneezing on it, or breathing on it, coughing on it, and potentially contaminating it. These are the swabs. These are 6 inches long. It's basically a Q-tip. It's sterile. This is what you will use to collect the DNA. I've got these little 2 milliliter vials that you would break off your swab, your applicator swab, into with your alcohol that you injected with your pipette. And the reason that you'd want to use something like this is because you only have 1 bottle of alcohol. Now what happens if the lip of your bottle of alcohol is touching every vial that you sample something with, and everything you have is being dipped into it? There's a good chance for cross-contamination, even if they're different samples. So something like this, which is basically a glorified eyedropper, would be used once and thrown away. Everything you have, everything that I have, is designed to be used once and then thrown away. Never use it again. I don't clean anything. I don't have 1 scalpel that I use, 1 set of tweezers. Everything's disposable. Get rid of it because I don't want to risk cross-contamination. Now, collecting the sample. If I have a pile of Bigfoot crap, I think it's Bigfoot crap, and I want to collect DNA from it, where is the DNA present on that pile of excrement? It's on the surface. So if I stab it or grab a lump of it and throw it in a bag and seal up the bag, what have I done? I haven't collected anything. Moisture, environmental insult, time are enemies of the DNA molecule. They will destroy it. So you're all proud of your find to get it to where it needs to go, and there's not going to be anything there for them to examine. Nothing viable. It will come back inconclusive. There's nothing there for them to examine. A lot of times on scat, you'll see sort of a shiny surface, a mucousy membrane around the scat. that mucousy membrane is where the DNA of the donor of that excrement is. Epithelial cells. Those are the cells that cover your body. Your skin, the lining of your intestine. That's where the Sasquatch's, if that's what it is, DNA is going to be present on that scat. If you stab the scat sample and break it off in your little vial, store it away, you just got a blueberry sample or grass or whatever it ate, not the animal that made it. So, you can moisten the tip of your swab with the alcohol or sterile water, and you roll it gently over the surface of that scat, making sure you get any of those nice reflective mucousy layers, because that's where the DNA is going to be. Collect it with your swab with a gloved hand. Put it in your vial, which you've already put alcohol in with the pipette that you used once, threw away. You can break off the tip. That's why I use the wooden ones. In the vial, seal it up. You can label it. And I went so far as to find pens that write on plastic, and it's permanent. They don't come off. And they're special pens. So you don't lose that data. You would then record that in your field book, and I'll discuss later field journal and other note-taking techniques. But that's where the DNA is going to be, on the outside. If you collect an entire scat sample, it can be, there won't be any DNA in it, most likely, that can be, what the donor was, determine what the donor was. But there are examinations that can be done as far as what was it eating, which that could be interesting. I imagine it would probably look very similar to what a bear eats, the other large mammal that lives in the same places that Sasquatch is reported to live. I discussed that. This is important. When you're swabbing the scat, you want to avoid getting hair or any other foreign material that might be on the scat. You don't want to include that in your sample. How many of you guys have seen coyote scat on a trail? What's it full of? Hair. A lot of times it's full of hair from the animal that it ate. So if your DNA sample consists of that hair, it's going to come back as deer, possum, raccoon, whatever it ate. So you want to make sure the surface and that mucousy layer, that's where those epithelial cells are going to be trapped. If it's a Sasquatch, that's where he is. And I discussed a little bit about the medium that you want to use to preserve it. Now, alcohol, there's actually a study done. These were marine biologists. They wanted to know how long would coral polyps be viable as far as DNA preserved in alcohol. Because when they go out to conduct their research, they're out on a boat for months. So if they find something, they can't race it back to a lab and have it examined. They might have to preserve it for weeks, months. So they actually compared preserving DNA in alcohol versus some other mediums, which I don't think you can get. You can make it. I found a recipe for it, but you need a lab for it to do that. And they reckoned that you could preserve a DNA sample that would be viable in alcohol at 200 proof is what they were talking about. But this is darn close, for about 6 months. So if you found something, reckon you have about 6 months to find somebody or save up the money to get it looked at. Not very many people have a refrigerator that will go minus 20 degrees Celsius. We can't do that. You certainly can't do it out in the woods in your truck. I can't. But with the alcohol, you can preserve it. You want to keep it out of the light. You want to keep it in a dark, cool place, as cool as you can find. And I talked a little bit about preservation. DNA is a very stable molecule, but it's susceptible to degradation and eventually rendered completely unviable if it is not stored in cold, dark, and dry conditions. If stored properly, this molecule can be preserved almost indefinitely. Properly, like I said, would be a freezer that goes 20 degrees Celsius. Nobody has that. So if you can keep it viable for 6 months, a year, hopefully that's enough time to get it to where it needs to go to have it looked at. And there are places that will do that. And I suspect they would especially do it if you didn't tell them it was a Sasquatch. If you call up a place and you go, I got some Squatch crap I want you to look at and prove that it's a Sasquatch. I already know it is, but I need to put it on my Facebook. They're probably not going to do it. If you find something, ask them to examine it because you suspect it's a bear, and they look at it thinking it's a bear, and it comes back as something else that they weren't expecting. That might be a better way to get an answer. I certainly wouldn't come out with Bigfoot, because